Okay, so we'll start now. Others can join me in well. Uh, so, just a second, yeah. So, welcome everyone. Today is the seventh problem solving session for the course Experimental Biotechnology. The course instructor for this course is Professor Vishal Trivedi from IIT Guwahati, and I am Shrija Ghosh, a PMRF from IIT Kanpur. I am here to take these live sessions to solve any kind of doubts you have regarding your assignments. So, today we will be discussing uh, antibody generation techniques and then different types of immunoassays where we'll use antibodies to detect different kinds of proteins or measure the titer of proteins. Okay, so till now in the previous classes we have discussed about electrophoresis and chromatography. So now this is a new technique that we will be discussing. So there will be multiple types of techniques under this criteria, and we'll go through each and every technique through solving different kinds of questions. Do let me know if you have any kinds of doubt in between the question. You can either stop me or you can ask your questions in the chat box, and I'll try to clear them up. Okay, so I'll start now. So starting with the first question, question number 1: which of the following is involved in humoral mediated immunity? Option A, T cells; option B, B cells; option C, antibody; and option D, plasma B cells. Which of the following is involved in humoral mediated immunity? Anybody knows the answer for this? If you already know the answer, you can write in the chat box, and then we can have a discussion if you are right or wrong. Any one of you wants to answer? Yeah, we have one answer, option B, B cells. Okay, anyone else? So the correct answer for this question is actually option C, antibody. So we have two types of immunity. One is humoral mediated immunity, the second is cell mediated immunity. Okay, so humoral mediated immunity and cell mediated immunity. In humoral mediated immunity, all the antibody mediated immunity comes under this criteria. Okay, and all the cell related immunity, so the immunity that arises from the T cells and B cells, they are basically the cell mediated immunity. B cells are the source of antibody, but the immunity that arises directly from the B cell is comes under cell mediated immunity. Okay, so we have T cells and B cells. These are basically our immune cells. Okay, so the immunity that arises directly through these cells, so there are certain mechanisms through which immunity can be developed. So what do you mean by immunity? Actually, it's our body's way to recognize different kinds of foreign materials that are not belonging to our body, and providing a defense mechanism against these foreign antigens or foreign particles. So anything that is not part of my body will be considered as foreign. Okay, so if I am exposed to some pathogen, some kind of infection, so any infectious bacteria, that bacterial particle will be considered as a foreign particle for my body, right? So once that bacteria, once that antigen, so any foreign particle is considered as an antigen for my body that can elicit an immune response, that can trigger an immune response. Okay, why this immune response will be triggered? Because my body now knows that this is not normal, this is not part of our system, and we need to defend this, because this can potentially cause infection or it can harm my body. So my body kind of defends itself. It's like if a thief enters your house, you will try to defend it, you will try to call the police, or you will try to fight it depending on your response system, right? So similarly, if a pathogen enters my body, it will be considered as a thief, because it is now going to harm my body. So my body will start defending that pathogen. How? By certain mechanisms that our body has already established inside it. So T cells and B cells are immune cells that have the capability of producing certain how do I say? So T cells and B cells are these immune cells. So they have the capability to elicit this immune response, to make a defense mechanism that can actually prevent our that can actually help our body to prevent these pathogenic infections. Okay, so sometimes these antigens are presented on these T cells, and then certain other cells that can know that okay, these cells are harmful, and then they can degrade those cells. So different kinds of processes. So generation of antibody is one such kind of process, and the immune response that is elicited after the antibody production. So what these antibodies do, they are generated against a certain antigen. Okay, so let's say a bacterial pathogen entered my body. Now my body knows that this is not normal, we have to kill this thing, and my body will generate cell mediated immune response and later humoral mediated immune response. So in the humoral mediated immune response, my body will generate certain antibodies. Those antibodies will be basically like memory cells. Like if again the same pathogen enters my body, those antibodies will directly bind to that pathogen and trigger a immune response, so that those pathogens are killed before causing any kind of infection. So antibodies basically are kind of a memory. Like if a thief enters your house and you know the face of the thief, if the same thief enters again, you will already know before you see that thief stealing something, you will already know that okay, this person was a thief, I have already seen him. So it's like that. Okay, so I hope this is clear. So antibody is part of humoral mediated immune response. T cells and B cells are part of cell mediated immune response. Okay, is this clear to everyone? Anybody has any kind of doubt regarding what is immunity, what are immune responses, and what are cell mediated and humoral mediated immune responses? So this is part of basic immunology. So this is the basis of how antibody is generated in the lab and how we are using it in different techniques while we are studying different kinds of proteins. Okay, so this immunology is not a direct part of this course, but then for doing any kind of biotechnology, you need to develop a base of immunology if you are going to use antibody based assays that are frequently used in biotechnology. Okay, any doubt? This is clear to everyone? Great. So I'll move to the next question now. Question number 2. The Y-shaped structure of an antibody is composed of how many protein chains? Option A 1, option B 4, option C 3, option D 2. The Y-shaped structure of an antibody is composed of how many protein chains? So you have to tell the number of protein chains in the Y-shaped structure of an antibody. If you know the structure of an antibody, you should be able to answer this question. Anybody wants to answer? Even if you don't know the answer, take a logical guess. I have already mentioned it's a Y-shaped structure, so take a logical guess how many chains can be there. Okay, we have one answer, option B. Number of chains is 4. Okay, anyone else wants to answer? So B is the correct answer. The Y-shaped structure of the antibody is composed of 4 protein chains, out of which 2 chains are called heavy chains and 2 chains are called light chains. So this is the structure of antibody. So here you have the antigen. So you have the antigen here. So we have in the Y-shaped structure, we have 2 heavy chains, the blue ones shown in blue color. These are the heavy chains, and then 2 light chains associated with the heavy chain through these disulfide bonds. And the top part of the Y, the open part of the Y, basically has the antigen binding site. So each antigen will be each antibody will be specific for a particular antigen. So you can see the binding is kind of like a shape complementary binding. Okay, so a complementary binding will happen between the antibody and the antigen at the antigen binding site, and this is the effector region. What do you mean by effector region? Basically, this part is going to bind to the antigen and elicit the immune response. This part is basically the one that will help in eliciting that immune response. So this part is for binding. This part will recognize, okay, things have bound here, and now basically signal mediation will happen from the effector region. Okay, so this part is called the FC region of the antibody, that is the constant region, and this is the Fab region, that is the antigen binding fragment. Okay, so antigen binding region and the constant region. So why it is called constant? It is almost conserved throughout all the antibodies. The amino acid sequence in this region is kind of conserved in all the antibodies, but the amino acid sequence in these regions will differ, obviously, because each antibody is specific for a different antigen. Okay, so for simplified purposes, if you are ever drawing a diagram of this, if you are ever drawing a, like if you are making a process in which you have to show antibody, we generally make the antibody like this, Y shaped with 2. So these are the 2 heavy chains and then 2 light chains outside the Y. So this represents the antigen binding sites, and this represents the FC region. All these chains are linked with disulfide bonds. You can see here disulfide bonds. So if you want to break these bonds, you can use any kind of reducing agent that will reduce these disulfide bond into SH groups, right? To break this bond, any kind of reducing agent, beta mercaptoethanol. You have studied about beta mercaptoethanol and DTT when we were discussing SDS page electrophoresis. Okay, so those substances, those chemicals can actually help reducing these disulfide linkages. Okay, and this part is called the hinge region because this is kind of flexible for the binding of the antigen. So based on the antigen, it can actually be little flexible and facilitate the binding of the antigen to the antibody. Okay, so we have the heavy chain and the light chains. So 4 chains, 2 types of chain, heavy and light, and 2 heavy chain and 2 light chains. The total number of protein chains is 4. This is clear? Any doubt in the structure of antibody? Okay, then I'll move on to the next question. Question number 3. In which phase of the antibody production there is entry of pathogen in the body and contact with immune cells? In which phase of the antibody production there is entry of pathogen in the body and contact with the immune cells? Option A, plateau phase. Option B, log phase. Option C, lag phase. Option D, decline phase. Which phase of the antibody production there is entry of pathogen in the body and contact with immune cells? Okay, we have one answer, option C, lag phase. Okay, anyone else wants to answer? Everybody agrees with option C then? So option C is the correct answer, lag phase. So we have 4 phases: lag, log, plateau and decline phase for during the antibody production. So during the lag phase, which is the first phase, that we have the antigen, like basically introduction of the antigen to the immune cells. Then our immune cells will recognize those antigen, produce the antibody. Then there will be a point where the antibody production will be maximum, and then there will be no further increase, and then the decline phase of the antibody production will start. So this is how the 4 phases of antibody production happens. So first phase, that is the lag phase, that the entry of the pathogen is happening and contacting with the immune cells. Now the immune cells, once they recognize that okay, this is an antigen, this is a foreign particle and we have to develop antibodies against it, there will be a steady rise in the antibody production in the log phase. So it's called a log phase because there is a logarithmic increase in the antibody titer or antibody concentration. In the plateau phase, it's kind of a stationary phase where the amount of antibody synthesis and its catabolism, that is its decline, is in equilibrium. So the number of antibodies that are produced is equal to the number of antibodies that are catabolized or that are degraded. Okay, so that is the plateau phase. So there is no extra antibodies being produced, and there is a stagnant stationary plateau that we see in the graph. Then starts the decline phase where the antibody titer starts decreasing again, because now the body has fought with the pathogen and the body does not need antibodies against those pathogens, and it starts decreasing in the bloodstream, in the serum. Okay. So this is actually the principle behind vaccination also. So I'm assuming all of you have gotten COVID vaccine, right? So this was the basis of, like, whichever vaccine you took, this was kind of the basis of the vaccination, the principle behind the vaccination. So in the lag phase here, the antigen is introduced. So whatever vaccine you have taken, that, consider that as the antigen. Okay. And then days after, so this is the number of days after immunization. So after few days you will see the log phase, where in your body the antibody against those particular antigens were being developed. And that is why, if I'm a, like, if you know, most of the people have gotten some kind of fever or some kind of ailment after, immediately after getting the vaccine dose, right? So did you all get the fever or anything after getting the vaccine dose, if you remember? So that was because of the increase in the antibody concentration. So antibodies, when they were increasing in our body, they were kind of fighting with the antigen that we have injected in our body. So after that, the plateau phase will reach, and once my body has recognized, okay, this is a pathogen and antibodies have been made against it, there will be a decline in the antibody phase. There comes the second dose. So all the vaccines had 2 doses initially, right? So those 2 doses are basically to give extra immunity, so that the body remembers that, okay, this is the pathogen, and whenever we are getting attacked by the real pathogen, my body will fight against it. Okay. So it's like, I am deliberately injecting few viral particles or viral antigens, so that my body recognizes, okay, this is a virus and we have to fight against it. So when I actually contact COVID virus, when I actually get in contact with the pathogen, the viral, the whole viral particle, the infectious particle, I won't develop any infection, because now my body knows, now my body has the antibodies and the ability to produce antibodies against such a infectious pathogen, and my body can fight against that pathogen without causing any infection. Okay. So that is the, this is actually the mechanism behind vaccine, like principle behind vaccination, how vaccine works. So this is true for all kinds of vaccine, whether it is polio vaccine, hepatitis vaccine, or COVID vaccine. Okay. So in the lag phase, that is the first phase, we have the entry of the pathogen and contact with the immune cells. Any doubt in this question? This is clear how the antibody production is happening inside the cells. So this is also important, because we are now going to use this method to actually produce antibody that we can later purify and use it for our other assays. Okay. So if I want antibodies outside the cells, you can simply inject a particular antigen in the rat or mouse, and then take its blood. Once the antibodies have produced inside the mouse, you can take the blood of the mouse and then isolate the antibodies for that particular antigen that you have injected, and then use those antibodies for any purpose that you want. You can use it for purification. If you remember, in the affinity chromatography, we have talked about antibody based affinity. So you can generate antibodies, and then you can fix those antibodies on a solid matrix, and then use it for purifying particular protein which you have used as an antigen to immunize the mouse. Okay. So any doubt in this part? So this is, I hope this is pretty clear. We have discussed antibody generation in the previous session also, so this is kind of continuation of that. Now we are going to see other applications of the antibody in the experimental biotechnology assays, different kinds of experiments that we can do in biotechnology. Okay, moving on to the next question. Question number 4. Which of the following is antigen presenting cells? Option A, macrophages. Option B, T cells. Option C, activated B cells. And option D, plasma cells. Which of the following is an antigen presenting cell? Macrophages, T cells, activated B cells and plasma cells. Okay, we have 3 answers. Option A, yes, that is the correct answer. So option A, that is macrophages are the antigen presenting cells. What do we mean by antigen presenting cell? By the name itself you can understand that these cells are presenting the antigens to the immune cells. So I have told you we have B cells and T cells that are the immune cells. So macrophages are basically the servers of these antigens. So whenever the antigen enters the body, macrophages will present these antigens to the T cells and B cells so that they can recognize, okay, this is an antigen, and then they can do their function. So this is how things kind of work. This is a diagrammatic representation where the antigen is there, and macrophages, they have certain receptors, certain cell surface proteins that have the capability of presenting those antigens. It's like a waiter serving your food in a hotel. Okay, so in a restaurant, if you are going and a waiter serving your food, so you are not going to take the food directly from the kitchen, right? You need someone to serve that food to you in a restaurant. So it's like that's kind of the same function macrophages are doing. They are taking the antigen and presenting it to the T cells. Now the T cells can take the antigen, and then later different kinds of immune responses are there. Here we have one kind of immune response where the T cells will kind of tell the B cells that, okay, this is the antigen, and then B cells will generate 2 types of cells, memory cells and plasma cells. Memory cells are the ones that I was talking about, where once you are immunized with certain kind of antigen, your memory cells will remember that antigen, and whenever you are attacked by the same pathogen containing those antigens, your memory cell will recognize that as a pathogen and kill it. Plasma cells, where on the other hand, plasma cells are the cells that are directly producing the antibodies. Okay, so this antibody will then mediate the humoral immunity. Okay, so this is just one kind of representation. There are other types of antigen, there are other types of immune response processes, but that is not part of this course, that is beyond the scope of this course, so we will not go into the details of that. But macrophages are basically the antigen presenting cells that present the antigen to the T cells. Okay, so all of you answered, most of you answered correctly, so I am assuming that you have you are clear about this question. Still, and if anybody has any doubt, you can write in the chat box. Otherwise, we will move to the next question. Question number 5. How are monoclonal antibodies produced? Option A, by collecting antibodies from different animals. Option B, by stimulating a single B cell clone and fusing it with myeloma cells. Option C, by isolating antibodies from a mixture of different B cell clones. And option D, by cloning T cells and generating hybridomas. How are monoclonal antibodies produced? Option A, by collecting antibodies from different cells, different animals. Option B, by stimulating a single B cell clone and fusing it with a myeloma cell. option C by isolating antibodies from a mixture of different B cell clones, and option D by cloning T cells and generating hybridomas. Okay, 2 people have answered option B by stimulating a single B cell clone and fusing it with myeloma. Okay, one more person is agreeing with option B. So yes, that is the correct answer. So first we will discuss what is a monoclonal and polyclonal antibody. What do we mean by monoclonal antibody? So option D is the correct answer. Stimulating a single B cell clone and fusing it with myeloma cell can actually help us produce monoclonal antibodies. But what do we mean by these monoclonal and polyclonal antibodies? So whenever I am talking about a pathogen, so any pathogen will have different kinds of antigen, or if I like, let's say just a second. So let's say if I have a viral particle, so it will have different kinds of proteins expressed on its cell surface. So each protein or each cell surface molecule can be used as an antigen. So we can't use the whole virus particle as the antigen because it is infectious in nature. So we will kind of inactivate this viral particle, or we will use a part of the virus to present it to cells as an antigen, like, look, this is a viral particle and you have to fight this. Okay, so now if I take a part of this virus, so now this is the antigen that we have. Now that antigen will have multiple epitopes against which this antibody can be produced. So even an antigen, a part of the viral particle or the bacterial particle, will have multiple epitopes, and each epitope will be different in nature. Okay, so here we have represented it in different colors. I am making it in different shapes, but it's just a representation. It's just for the representation purpose. But each epitope means a part of that antigen now. So each part of that antigen will have different kinds of properties. So I have told you the antibody, the antigen binding site of the antibody. You have studied that Y-shaped structure, right? So I told you this was the antigen binding site. So this part is very specific for particular epitope. So if antibody has been produced for this epitope, that antibody will be different. If an antibody is produced for this epitope, that will be different. So for each epitope, the antibody generated will be different. So when we are immunizing a mice, let's say we have a mouse and we are injecting the mice with the antigen, right? And just taking the this side arrow, and then just taking the blood of that mice and separating the serum from the blood and purifying the antibodies, right? This is the general process that we have studied in the previous session also. So this, when we are doing this serum has different kinds of cells, right? So different cell will produce different types of antibodies, like different types of antibodies, as in different antibodies for different epitopes. All the antibodies will be specific for this antigen, but they will not be particular for a single epitope. So these are called polyclonal, mixture of different epitopes, mixture of different antibodies that are specific for different epitopes. That is a polyclonal antibody. Why clonal word is used? Because now serum is having all the B cells, the the B cells are producing the antibodies, right? So all the B cells will have different, so these are cells, right? So each cell will be producing different antibody. Each cell is going to produce different antibody, right? So now what we are doing, we are going to pick a single cell and make antibodies. So that's how we can get monoclonal antibodies from one cell clone. Okay, so from one cell we will make multiple cells. So that will be a clone of that particular single cell, and that clone, that single clone can give single type of antibody against a particular epitope. So that will be more specific in its binding efficacies. Now polyclonal antibodies, the binding can differ based on which kind of epitope is present. So let's say like if we have monoclonal antibodies, since all the antibodies are going to bind to a single epitope, the binding efficacy, the kinetic of binding will be same for each and every molecule, but here it will be different. Okay, so that is the difference between monoclonal and monoclonal and polyclonal. Sorry, just a second. So that is the difference between monoclonal and polyclonal antibodies. Now we have in the question, how are the monoclonal ones produced? So you have answered by stimulating a single B cell clone and fusing it with myeloma cells. So that is a process which is also known as a hybridoma technology. So what we do, we inject the mouse with the antigen and take the blood, and then we take the spleen cells out of these mouse, and we fuse these spleen cells with the myeloma cells. So now these myeloma cells are a mutant which lack the ability to synthesize its own DNA. So when we are doing fusion and when we are doing the selection, only the fused cells can survive. The cells that are not fused, the only spleen cells or only myeloma cells are actually selected out in the selection media, and then once we have the fused cell, from that we can pick single cells and see which of the cell is producing the antibodies with higher titer and desired function, and then we can purify the antibody using that cell, and we can purify the antibody using any of the chromatographic techniques that we have discussed. So this is how we generate monoclonal antibodies using the hybridoma technology, by taking out the spleen cells, by cloning single B cell, by fusing single B cell with the myeloma cells for making the hybrid cells. These are called the hybridomas. The separate hybrid cells are called the hybridomas. Okay. So this is clear to everyone, hybridoma technology. Okay. Moving on to the next question, question number 6. Hat medium inhibits dash. Hat medium inhibits dash. Option A, nucleic acid synthesis. Option B, cell membrane synthesis. Option C, antibody synthesis, and option D, protein synthesis. Hat medium inhibits dash. Which of the following is inhibited by hat medium? Nucleic acid synthesis, cell membrane synthesis, antibody synthesis, and protein synthesis. Anybody knows the answer? Okay, I'm getting one answer, option A, nucleic acid synthesis. Anybody else wants to answer? So nucleic acid synthesis is the correct answer, option A. So HAT medium is the medium that I was talking about, that is used for selection of hybridomas during the process of making monoclonal antibodies. So it's the hypoxanthine aminopterin thymidine medium. So H stands for hypoxanthine, A stands for aminopterin, and T stands for thymidine. So what these cells do, what this media do is it has aminopterin, right? So aminopterin can inhibit the salvage path, de novo pathway of the DNA synthesis. So DNA synthesis has two pathways. Nucleic acid synthesis has two pathways. One is de novo pathway and the second one is salvage pathway. Okay. So in the de novo pathway, everything is synthesized from the new components, like we will provide the raw material thymidine. Just a second. So we will provide the raw material thymidine and adenine. So all the ATGC will be provided, and the cells will synthesize the nucleic acid from the scratch. In the salvage pathway, what is happening? Any kind of materials that are going to be waste, from that what our body does, it kind of breaks it down into nucleic acid components, the individual components of a nucleic acid, and resynthesizes the nucleic acid from those pathways. So it's kind of it is salvaging the whatever present inside the cell. And de novo means it is making something from the scratch, it is making something new. So de novo. Okay. So now in the de novo pathway, if I add aminopterin, it is kind of inhibiting that de novo pathway. Okay. And the salvage pathway is already blocked in the myeloma cells. So in this I've told you that spleen cells and myeloma cells are fused, right? So these myeloma cells are basically a mutant in which salvage pathway is absent. Okay, so it is either HGPRT mutant or thymidine kinase mutant. So two types of mutations can happen, thymidine kinase or HGPRT. So both of these mutants can cause loss of function in the salvage pathway, and aminopterin is blocking the de novo pathway. So you can understand the myeloma cells cannot survive on its own. So when we are doing this fusion, all the myeloma cells that will be there which have not fused, all these unfused myeloma cells, they will basically die because they can't produce its own DNA. Without producing its own DNA, cells cannot divide, so they will kind of die. And the B cells, the spleen cells that we have here, they can actually produce the nucleic acid, but since they are the primary cell lines, they are not immortal. Only cancer cells are immortal, they can keep on dividing without dying. But the mortal cells, the primary cell lines, they have certain lifespan, they will grow till that lifespan and then they will eventually die. So since they don't have any immortality in them, they will anyway die if they are not fused with the immortal cells. So when we have the fused hybrid, where we have the myeloma cells and the spleen cells, the hybrid now basically is having the immortality, so it is not going to die, and it is also having the salvage pathway that is naturally present in all the cells. So it can actually grow in the HAT medium where the de novo pathway is inhibited, salvage pathway is there. So the immortal myeloma cells are now using the salvage pathway of the B cells, because now it's a hybrid, right? So whatever is there in the B cells can be used by the myeloma cells, and whatever is there in the myeloma cells can be used by the B cells. So it's a fusion. So they can remain immortal by synthesizing its own DNA, and since it's a cancer cell, it can keep on growing. So that's how we can select the cells that are surviving on the hat medium are the fused hybrid. So that's how the selection between the hybrid, like selection for the hybridomas are done. And then once you have the selected hybridoma, you can pick a single clone by doing serial dilution, and then you can get the cells from a single clone. You can get the antibodies from a single clone that will be known as the monoclonal antibodies. This is clear to everyone? Anybody has any doubt till now? Till now, whatever we have discussed, if anyone has any doubt, please write in the chat box. Meanwhile, I'll move to the next question. Question number 7. Which of the following is commonly used as a carrier particle in the indirect hemagglutination reaction? Which of the following is commonly used as the carrier particle in the indirect hemagglutination reaction? Option A, red blood cells, option B, white blood cells, option C, bacterial cells, and option D, platelets. Which of the following is the carrier particle in indirect hemagglutination reaction? Option A, red blood cells, option B, white blood cells, option C, bacterial cells, and option D, platelets. Anybody wants to answer? Yes, we have two answers. Shantan and Megha are answering option A, red blood cells. Okay. Anyone else wants to answer? So option A is the correct answer here, red blood cells. So red blood cells are the carrier particles in the hemagglutination reaction. So what is hemagglutination? Basically, we have the red blood cells. We can actually add viral particles to these red blood cells. So if the viral particles are there, we will see the red blood cells getting clumped, and these clumped red blood cells will now settle down very slowly as compared to the red blood cells that are not forming a clump and that do not have a virus. So once you take a blood sample from an infected patient and blood sample from the healthy patient, you can see the blood having like if you take the blood in a petri dish or a well plate, you can see a red dot forming from the top. When you will see, you can see a red dot forming in these cells that do not have any virus. The ones that have the viral particles, since the red blood cells are getting agglutinated, because of the agglutination, it kind of appears dispersed in the wells, and it does not show a single red dot. So this is the picture of a 96 well plate where we have shown the agglutination. So you can see these red dots. These are basically showing the healthy cells, and the ones that do not show these red dots basically have the viral particles, and they are causing the red blood cells to form a clump that is not settling down fast enough. So we do not see the red dot. Okay. So you can do these experiments in a serial dilution manner, where you can keep on diluting the samples and add to the red blood cells, and then see if the agglutination is happening. So till what point of dilution the agglutination starts, agglutination occurs, basically shows the titer of the virus that is present. So if the virus is present in a higher quantity, you will see that till the higher dilution also there won't be any red spot, there will be agglutination occurring. Okay, any doubt in the previous question? Clear to everyone? Okay. Okay, if no one is asking anything, I'm hoping that you all have understood this. So I'm moving to the next question, question number 8. Which of the following diseases can be diagnosed using the indirect hemagglutination reaction? So I've just now discussed what is indirect hemagglutination reaction. So which of the following diseases can be diagnosed using this technique? Option A, influenza. Option B, bacterial infection. Option C, malaria. And option D, Lyme disease. Okay, we have two answers. Option A, both of them are option A, that is influenza. Anyone else wants to try? Okay, so option A is the correct answer. So generally these indirect hemagglutination reactions are used for detecting the viral titer in the sample. So influenza is caused by a virus, so influenza can be diagnosed using the indirect hemagglutination assay. Okay, anyone having any doubt? Everybody understood how the indirect hemagglutination assay is done? What is the agglutination reaction? Great. Okay. So I'll move on to the next question now. Question number 9. The Octoloni double diffusion and double immunodiffusion technique is used to option A, determine blood type. Option B, detect specific antigen in the sample. Option C, quantify antibody concentration. And option D, purify the antigens. The Octoloni double immunodiffusion technique is used to option A, determine blood type. Option B, detect specific antigens in a sample. Option C, quantify antibody concentration. And option D, purify the antigens. Okay, we have one answer, option B. Anyone else wants to answer? Everyone agrees with option B then? Okay, so option B is the correct answer. Detect specific antigens in a sample. So Octoloni double immunodiffusion technique is a technique where we add the antibody, and we can add different types of antigens, and then based on their reactivity we can actually detect a specific antigen in a sample. So this is a Octoloni double diffusion technique plate. So what we do is basically we take normal agar plate. So we mix agar in the water, boil it, and then we pour it in the petri dish, and we make wells like this. So we can use a cork borer to make a well inside, basically make a hole inside these agar, solidified agar, and then in these wells carefully we can use a micropipette to add the antigen and antibody samples. So this well will have the antibody, and all these wells will have different types of antigen. So let's say antigen 1, antigen 2, 3. So now I have added different antigens in different wells, and we have the antibody in the center. So now, since this is a agar plate, whatever liquid I am putting, these antibodies and antigens, they can diffuse in the plate. So they can diffuse radially in the plate. So the antigens are diffusing from these wells, the antibody is also diffusing out of this well. Now if this antigen is reacting with this antibody, that means if the antibody that I have added, that I have taken, is binding to that particular antigen, then there will be a precipitation. Why there will be a precipitation? Because you have studied about agglutination, right? So because of the agglutination, the antigen and antibody will make a complex, and there will be a precipitation, and that line of precipitation can be seen in the agar plate. So it will be seen as a whitish line in the transparent agar plate, like you can see in this figure. This is exactly how we see it when we are doing the experiment also, and this line actually shows that this antibody is reacting with antigen 6, it is reacting with antigen 4, but it is not reacting with antigen 1 and 2, because there is no reactivity with the antigen 1 and 2. Now this line can be read as read in 3 different ways. So if I have just 2 antigens, let's say if I am doing this in a smaller setup, and I have the antibody here and 2 antigens here. So if I get a line like this, that means both of the antigens are reacting with the antibody in the same manner. Okay, but if there is a overlap between these lines, so we have the antigens are in the top wells, and then antibody is in the center well, and if we have something like this, that means there is some kind of cross reactivity between the 2 samples. And if we have a line like this, that means there is a partial similarity between these 2 antigens. So that's how we can actually see what kind of interaction is there. We can detect a specific kind of antigen. So if I know that this antibody is specific towards a certain protein, and I have added 6 antigens here, I can actually detect a specific, okay, this antigen belongs to the protein of interest. Okay, so to check which antigen is reacting with the particular antibody, I can use this technique, and this is a very simple technique. You just need agar and the antigen antibody samples. You don't have to do a lot. You just have to pour the agar once it is solidified, make the wells, pour the sample, like add the samples in the well using a micropipette, and incubate it to see the results. It's very simple technique, and it is very effective technique. It is a very fast technique to see whether the antibody is reacting with the antigen or not. This is clear? So double diffusion technique is clear to everybody?
Hello, ma'am.
Yeah.
Ma'am, I have a doubt. If there is one antibody and one antigen,
Yeah.
then how will we recognize the binding by this precipitation you told us?
If there is one
And
antigen and one antibody,
yes.
so if they are if the antigen is binding with the antibody, you will see
Mm.
the immunodiffusion line, because the antigen, if the antigen antibody binding is happening, there will be that precipitation that can be seen in the agar plate. So the agar plate is transparent, so if there is a precipitation, you will see a whitish line.
Between both the antigen and antibody, right?
Yes, between the antigen and antibody. So here in this figure also, if you see, the
Mm.
center well has I'll just remove the ink.
So in this antigen 1 and antigen 3 जो अभी आपने बताया
Yeah,
था.
yeah.
Yeah.
So
So
this
they
is,
have no role in binding to
so
the
no,
antibody.
so that is what we were checking. So if I if I have an antibody and I have
Mm.
6 different types of antigens, and I want to see which antigen is actually reacting with this antibody. So you can
Mm.
check it by doing this assay, and whichever antigen is reacting with this antibody can actually show the precipitation line, and you can see that okay, this particular antigen reacts with this particular antibody. You can do the same technique for a different antibody, again take a different plate and make 6 wells like this and add, and then you can see okay, this antibody is for this antigen, this antibody is for this antigen. Like that you can separate the antigens and the antibodies.
No, this is well understood that which antibody will bind to that, but
Yeah,
the confusion I am having is about this the precipitation line. I mean,
yeah.
it is connected to the antigen and forming a triangle, and मतलब how will we recognize कि to which it is connecting? It is वो वाली इसके साथ नहीं antibody के साथ bind नहीं करेगी. You getting my point?
So no, I'm actually not getting the point. So if we are getting a line, that means that antigen is binding to the antibody, right? So if I
Hmm.
have just two antigens, let's say
Hmm.
I have the antibody here, okay, and I have two antigens, 1 and 2. If I am
Okay.
getting a line here and not getting a line this side, that means second
Hmm.
antigen is not reacting with the antibody at all.
Okay.
If I am getting a line both side like this, that means there is the antigen 1 and antigen 2 are identical to each other, because there is complete like the triangle that is forming does not have any intersection. If there is intersection in this uh, precipitation line, that means there is non-identical antigen. So both of these antigens are actually binding to the antibody, but these antigens are non-identical to each other. So probably the
Okay.
antibody is polyclonal, all, or the both the antigens have different kinds of epitopes which are specific to this particular antibody, something like that. Okay, and if we
Hmm.
have a line like this, like if we have a well here, and then the antibody well here, if we have a line like this, that means there is a partial identity between these two antigens. Is
Okay.
that making it clearer? I am not getting your question actually.
Uh, no, actually, I am confused about this precipitation line. मतलब, but
So
now it's clear. No,
so
okay, I
if
got
you
it.
have like if you have just a single well of antigen and antibody, you will see a straight line between the antigen and antibody if they are reacting to
Hmm.
each other. That is
I
the
got your point.
only
Yeah,
thing.
ma'am, I got it.
Yeah, yeah. Okay, we have certain things in the chat box also. uh, if the antibody is specific to only one antigen among the others, then what will be the result? There will be just one straight line. I've just told you. I think now it is clear. And can you give again number of antigens, number to the antigen well? Yes. So I just randomly numbered it 1 2 3 4, 5 and 6, and this was the antibody. The center well has the antibody. So do you have any question regarding this, Bhavani? Are your doubts clear? Bhavani, can you unmute your mic and respond? Yeah, okay, great. Uh, and another question was from Sohar. Are you? I think that I have already explained, right? So now you understand how the antibody is specific to only one antigen. Like what will happen if the antibody is specific to only one antigen, right? Is there any other doubt? Great. Cool, then we'll move on to the next question. The next question is about radial immunodiffusion assay. So that concept will get clearer, I am hoping. So which parameter can be estimated by measuring the diameter of the precipitin ring in the radial immunodiffusion assay? Option A, antibody concentration. Option B, pH of the solution. Option C, viscosity of the gel. And option D, antigen concentration. Which parameter can be estimated by measuring the diameter of the precipitin ring in the radial immunodiffusion assay? Okay. Option A, I have one answer as option A, antibody concentration. Okay. Anyone else wants to answer? Okay, everyone is going with the antibody concentration. Just let me ask you one simple question. Why antibody and why not antigen? Whoever answered option A can, like one of you can explain what's your thought process. Okay, I'll explain the technique and then we'll come back to this. So the correct answer is antigen concentration actually. So I'm understanding that you might have understood the technique in an opposite way. So in this so this is very similar to the Octoloni immunodiffusion method. So in this technique what we do is we have the antibody in the agar plate. So when we are so let's say I have a flask and I have taken the agar solution. So agar solution, once I have boiled it and I let it cool down to like 50 degrees Celsius or so, where it is still liquid but not hot enough to degrade the antibody. At that stage, I'll just add the antibody solution to the agar and then pour this agar on a petri dish. Okay. Once the agar will solidify, now the whole plate has antibodies in it. Okay, the whole plate has antibody in it. And now what will I do? What I will make the wells in the similar way I was making the wells in the Octoloni immunodiffusion technique. In the same way we will make the wells, right? So once we make the wells, what we will do, we will add different concentrations of antigens, okay, known antigens. So let's say I have an antigen. Just a second, something is wrong with my pen. Okay, let's say I have an antigen and I am making serial dilution of that antigen. So I know the concentration of antigen, it is some xmg. Okay, So I have made different concentrations, so I have diluted that, so it becomes x by 2, x by 4, like that. So now I know the concentration of these antigens. I will add it in the well, let's say 1, 2, 3, 4 and 5. I have added that antigen in these 5 wells, 1, 2, 3, 4 and 5, and I have an unknown antigen whose concentration I want to know. I don't know the concentration of that unknown antigen and I want to know, and what I will do, I will add that unknown antigen in the 6th well. Now based on the concentration, now let's say the most concentrated antigen was added in the first well, so more amount of antigen, it will diffuse in the well, and the same precipitation principle will happen. So now the antigen is diffusing radially in the from the well in a circular way, and the plate has antibody, so the antibody is going to react with that antigen and make a precipitin ring. So the ring that you can see, this is basically due to the precipitation in the plate. So higher the concentration, more the diffusion will happen and the ring will be bigger. Okay, now in the diluted sample, like in the 3, 4, 2, 5, so these are basically kind of serial dilutions. Okay, so each sample will have a different size of ring based on its concentration. Higher the concentration, bigger the ring; lower the concentration, smaller the ring. Now what we can do, we can take a scale and measure the diameter of each of these rings. We can measure the diameter of each of these rings and make a standard plot. So what will happen, okay, my pen is not working, I'll just explain like that. So we'll make a standard plot where we'll have the radius of the or the diameter of each uh, precipitin ring versus the concentration. So concentration can be plotted on the x axis. So increasing concentration will have a increasing diameter of the ring. So basically you will get a linear curve. Whatever the concentration will correspond to a particular diameter. Now the unknown sample that we have loaded in the 6th well, we can just check the diameter of this. See in the plot, or from the equation of the plot, we can directly get the concentration of that unknown antigen. So this is the principle behind this is same as the octal only one, but for this we are determining the concentration of antigen. In that technique we were seeing the uh, different antigen reactions with a particular antibody. Okay, so different kinds of techniques can be used to check different properties. Okay, so this is used for the concentration determination of the antigen, not the antibody. Okay, so all of you have answered option A. Now I think like I hope this is clear now. And do you understand by what do I mean by the precipitation of antigen and antibody? Is this technique, both of these techniques, are these techniques clear now? If it is not clear, I can explain again. But ma'am, can't we spread the antigen and add the antibody in the well? We can do that, but if you spread it, there is a chance that it won't be uniform. First of all, that can be done. Second thing, depends on what kind of antigen you are using, what kind of antibody you are checking for. So that can be done, but I don't know how accurate that process will be. In this thing, you are mixing the antibody in the agar and pouring it, so it is kind of uniformly distributed throughout the plate. So whatever ring you will get, it won't be a false result. But when you're spreading the antigen, uh, if the spreading is not done uniformly, you can get false results for the ring diameter. So it has a technical issue basically, so that's why generally the opposite thing is done. I don't know if there is any other reason for doing the opposite thing, but mostly I have seen this, like this is a very common practical that people do when, like if you are doing a biotechnology course, this is a very common experiment that we do. So I am not sure why the second thing is not done regularly, but as far as I know, spreading it won't be uniform and it won't give you exact linear curve. Anyone else? Okay. Is it clear to everyone now? Just give me a minute, my PPT got stuck, I think. I'll just stop presenting and present my screen again. Just bear with me for 2 minutes. Okay. I hope my screen is visible. Moving to the next question, question number 11. The bait molecule used in a pull down assay is typically option A, a small molecule drug, option B, an antibody, option C, protein of interest, and option D, DNA plasmid. The bait molecule used in a pull down assay is typically a small molecule drug, an antibody, protein of interest, or a DNA plasmid. Anybody wants to answer? Pull down assay is a type of immunoprecipitation assay where we are using where we are using a particular bait molecule to capture a prey, we can say. So anybody has any idea, like, now, if using this hint, if you can understand what can be the answer? Yeah, option C, that is the correct answer. Very good. So protein of interest. So in the pull down assay, what we do, we have a GST that is kind of attached to a matrix, like we had the affinity, like we had an affinity chromatography. Similar to that, we have GST, that is glutathione S transferase, that is attached to a Cephirose or a agarose matrix, and in that we can add the cell lysate. Now we can have two types of GST with the bait, that is the protein of interest. Now this bait we are checking, this is the protein of interest, and we want to now check the other proteins that are present in the cell lysate. So cell lysate protein interacting with this protein of interest. Okay, so cell lysate protein interacting with this protein of interest. Okay, so now what we'll do, we'll take a bead with the GST with the bait attached to it, and we'll take control GST beads because we want to check if the so we'll take the GST beads without the bait to check whether there is any non-specific binding with the GST itself. Okay, then we'll take the cell lysate with the bait and without the bait, incubate it, and then spin it down, so everything will, so it's called a pull down assay. So when we spin it down, everything will settle down in the bottom, because these are heavy things. GST is there, bead is there, then your proteins will also bind to it, and bait is also bound to it. So it's a heavy complex. So when you centrifuge it, it will settle down. So that's why it's called a pull down assay, because you are pulling down your samples and then you can check the samples after centrifuging. So once you centrifuge, you take out the supernatant and check the pellet for your sample. So what you will do, you will run a SDS page gel. So you know, in the SDS page the separation is based on size. So what we'll do, we in the control you will see a GST band, since it's also a protein, it's an enzyme, so it will also show a band, and if there is any non-specific binding, those proteins will show. So this green protein is basically a non-specific binding protein. The things that have specifically bound to the bait, the protein of interest, I am interested in reading studying those proteins. So the purple and the red ones are the proteins that have actually bound to the bait. If they would have bound to the GST tag, you would have seen it in the control group also, but in the control group we only see the green one, that is non-specific binding. So we'll ignore the green one and we'll study the purple and the red one, red protein. So after that, whatever is your interest, so if you want to see the sequence of this protein, you can excise this band, take out the protein, and then put it in the mass spectrometer to check to identify the protein. And so there are other things that you can do with it, so that depends on your type of experiment. But then in this pull down assay, We basically take a protein of interest and see what all other proteins are interacting with this particular protein. So it is type of a immunoprecipitation. So in immunoprecipitation, instead of this GST and BAIT, we have an antibody. So in a solid matrix, we have an antibody. It's like the affinity chromatography, similar to that. You add the cell lysate, whatever will bind to the antibody will be bound, and then you can just spin it down to get the bound complex and run it over a gel. You will get an antibody band and you will get the bound protein band. Okay, so this is how immunoprecipitation or a pull down assay is done. Any doubt in this? Any doubt in this technique or in the question? Okay, so I'm hoping that it's clear to everyone. So I'll move to the next question, question number 12. The target molecule, that is the antigen, in a sandwich ELISA is captured between which two antibodies? Option A, primary antibody and the enzyme conjugated secondary antibody. Option B, blocking antibody and the substrate conjugated antibody. Option C, capture antibody and the detection antibody. And option D, secondary antibody and the tertiary antibody. So in the sandwich ELISA, the target molecule is captured between which two types of antibody? Primary and the enzyme conjugated secondary antibody, blocking antibody and substrate conjugated antibody, capture antibody and detection antibody, and secondary antibody and tertiary antibody. Which of the following option is correct? Option C, capture antibody and detection antibody. Okay, anyone else wants to answer? Okay, you are right actually. Option C is the correct answer, capture antibody and the detection antibody. So in a sandwich ELISA, basically we have the capture antibody on the plate, to which the antigen or the target molecule, we let the target molecule bind, and then over that target molecule bound to the capture antibody, we will add the detection antibody, and it will basically sandwich the antigen molecule between the capture antibody and the detection antibody. So here is the figure. So you can see there are 4 types of ELISA: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. So in the direct ELISA, we have the antigen coated plate. We can add the antibody, and the antibody will have some kind of fluorophore or some kind of detection technique, or some kind of substrate to which it can react to, and we can get the signal. We can check if there is signal. We can say that okay, the antibody is binding to this antigen, that means there is this antibody is reacting, or if like whatever we want to check. If we want to check whether the antigen is reacting to the antibody or whether the antibody is reacting to the antigen, that is like depends on your experiment. In indirect ELISA, we have the antigen coated plate. We have the primary antibody, and on that primary antibody has some specificity to a secondary antibody. So this secondary antibody, for this secondary antibody, this primary antibody is the antigen. Okay, think like that. So you have the antigen, then you have the primary antibody that is binding to the antigen, then you add the secondary antibody that has the fluorophore, or that has some kind of colorimetric detection technique, and once you add the secondary antibody and add the substrate, you will see the color, and you can see by seeing the color that okay, this particular antigen is reacting to the primary antibody. So if there is no reaction between the primary antibody and antigen, the secondary antibody will also not bind, because when we are doing ELISA, in each step we are doing a washing step. So we have the antigen coated plate. If we add the primary antibody, after adding the primary antibody, we will wash the plate so that any unbound fraction will get removed. Then we will add the secondary antibody. Again we will wash the plate for removing any unbound fraction. So if there is no unbound primary antibody, we will not see any secondary antibody reaction also. So if there is bound primary antibody, that means if the antigen is reacting with the primary antibody, only then we will see the secondary antibody reaction also. So that is indirect ELISA. In sandwich ELISA, instead of an antigen coated plate, we are having an antibody coated plate. Okay, and we are adding the antigen from the top. So we have the plate coated with the antibody, we are adding the antigen on the top, and then again we are adding a second antibody, that is a detection antibody, that will bind to that antigen. So this is kind of a sandwich, right? You have, like, these are two breads, and between that you have the filling, you have the antigen. So one antibody here, one antibody here, and you have the antigen in between. So that's it. This is the capture antibody and this is the detection antibody. Now detection can be done in two ways. Either you can have a direct ELISA kind of detection where you add the antibody that is having some kind of substrate specificity, or you can add a primary antibody and then a secondary antibody for making it more specific. So that depends, but this is kind of a sandwich. Then you have the last one is competitive ELISA. We will discuss about this in the next question. Any doubt in the three types of ELISA that have been discussed? Direct, indirect, and sandwich. Do you understand how ELISA is being done? Okay, I hope this is clear. Okay. Next question, question number 13. How does competitive ELISA work? Option A, the target antigen competes with the labeled antigen for binding to limited antibodies. Option B, the target antibody competes with the labeled antigen for binding to a limited antigens. Option C, the target antibody directly binds to the enzyme. And option D, target antigen directly binds to the substrate. How does competitive ELISA work? Anybody knows the answer? So by far you have understood what ELISA is, right? So Bhavani has answered option B. Target antibody competes with the labeled antigen for binding to limited antigens. Okay. Anyone else wants to answer? Come on, try to answer it at least. Okay. So the correct answer here is actually option A. The target antigen competes with the labeled antigen for binding to limited antibodies. So what happens in the competitive ELISA is you have the antibodies, you already add the antibody to your antigen solution. Okay, so make the antigen antibody complex in the solution. Now whatever remaining antibodies will be there, that will be added to the plate of the competitive ELISA plate, and then we will see. So here you can see, so you have the antigen on the plate. So whatever unbound antigen unbound antibody will be there, will react to this plate, and it will kind of compete with the inhibitor antigen. So we will add another antigen that will compete with this limited amount of antibodies that are added in the solution. Okay, and then we can have the detection. So that is competitive ELISA. So antibodies are limited here, and we add two types of antigen that are competing with each other for the binding. Okay, so this is clear. Any doubt in this? If you have any kind of doubt, you can ask me. I'm assuming everything is clear if you are not asking anything. Okay, if there's no doubt, then we can move to the next question. Question number 14. What is the purpose of membrane blocking in Western blot? What is the purpose of membrane blocking in Western blotting? Option A, to prevent proteins from binding to the membrane. Option B, to remove excess antibodies. Option C, prevent non-specific binding and reduce background noise. And option D, visualize protein bands. What is the purpose of membrane blocking in the Western blot? To prevent proteins from binding to the membrane, to remove excess antibodies, to prevent non-specific binding and reduce background noise, or to visualize the protein bands. Why do we do the blocking step in Western blotting? Yeah, Kanika has answered option C. Okay, another answer, option C. Okay, so option C is the correct answer. We have two people agreeing to it also. So it we do the membrane blocking to prevent non-specific binding. So what we generally do is, so in Western blot we are separating the proteins in SDS page, then we are blotting the protein bands on a membrane, generally a nitrocellulose membrane or a nylon membrane. Then on that membrane, before adding the antibody to see where the antibody is binding, we generally block the whole membrane by dipping the membrane in a BSA solution or a dried milk powder solution. So these solutions have other proteins, so they will kind of block the whole surface of the membrane. Then you can wash the membrane slightly and then add the antibody. So antibody will not have any non-specific binding, it will only give the signal where your protein of interest is there, okay, on the membrane. It won't give a lot of background. Otherwise what will happen, when you will see the image of the blot, you will see a lot of background blackish noise and your protein band, so it would be difficult for you to visualize the band. So if you do this blocking step, it would be easier. So you will see a clean background and a black band for your protein. Okay, so that's why we do membrane blocking. And this kind of blocking we do in other immunoassays also, wherever we are using antibodies to check the antigen reaction, we generally do this kind of blocking step. So in ELISA also we do blocking step before adding the antibodies, so that there is no non-specific signal that we are seeing. Okay, anyone has any doubt? Okay, moving on to the next question now. Question number 15. The reagent that is used to develop an HRP based secondary antibody system in the Western blot is option A BCIP NBT, option B substituted 1,2-dioxetane phosphate, option C dimazone, and option D luminol and H2O2. The reagent that is used to develop an HRP based secondary antibody system in the Western blot is. Okay, we have Saudra answered luminol H2O2. Yes, that is the correct answer here. Luminol H2O2 is used to develop the HRP based secondary antibody. So HRP is the horseradish peroxidase. So it can actually react. So here is a table of chromogenic and luminescent visualization of the HRP based and the alkaline phosphatase based antibodies. So if you remember in the ELISA, I have shown you here a green dot over the antibodies that is reacting to a substrate to give a product, right? So this green dot is basically the enzyme that is linked with this antibody. So the full form of ELISA is enzyme linked immunosorbent assay, right? So enzyme is linked to the antibody and that substrate reacts to that enzyme to give a product that will give a color or that will give a luminescence. Okay, that is the basic principle behind ELISA. So this enzyme, there are most commonly used enzymes, that is HRP, horseradish peroxidase, or alkaline phosphatase. So for visualizing horseradish peroxidase, you can use all these reagents. So these will act as the substrate. So if you use 4-chloronaphthol, it will basically give a oxidized product to form purple precipitate. So you will get a purple coloration and you can detect that purple color at a particular wavelength. Then if you use DAB nickel chloride, it will form a dark brown precipitate. So you can see the precipitate, and amount of precipitate will determine the amount of antigen antibody reaction conjugate. Then you can use TMB, that will form dark purple stain. For alkaline phosphatase, you can use BCIP and NBT, where BCIP hydrolysis produces an indigo precipitate after oxidation with NBT. And then there are luminescent techniques where you can use luminol and H2O2. So H2O2 is a hydrogen peroxide, right? So this will basically oxidized luminol gives off the blue light. So you will see a bluish light in this technique by using this substrate. For alkaline phosphatase, you can use substituted 1,2-dioxetane phosphate, where it will dephosphorylated. So alkaline phosphatase will basically remove the phosphate from this substrate, and it will give off light. Okay. So these are different kinds of substrates that can be used for different kinds of enzymes that are linked on the antibodies, depending on what is available and what is the method of detection. You can use any of these substrates. Okay. Any doubt in this? This is pretty straightforward. Anyone has any doubt? Okay. Moving on to the next question. The primary purpose of SDS page in Western blotting is option A, to separate proteins based on their size, option B, to transfer proteins to a membrane, option C, to detect specific proteins using antibodies, and option D, to block non-specific binding sites on the membrane. What is the primary purpose of SDS page in Western blotting? Yeah, Bhavani and Kanika both have answered option A. Yes, that is the right answer. So it is just logical. You use SDS page to separate the proteins, and then you can blot it on a membrane to visualize the protein. So I hope this is clear to everybody. We have discussed about SDS page a lot in the previous sessions to have an idea that this is pretty straightforward. I'll move on to the next question. Question number 17. Which of the following is used to detect the primary antibody in the Western blotting? Option A, a secondary antibody. Option B, a blocking buffer. Option C, SDS page buffer. And option D, transfer buffer. This is also simple. Which of the following can be used to detect primary antibody in a Western blotting? We have already discussed about ELISA, so you should be able to answer this. Come on, I want answers from you guys. Now we have discussed 16 questions before this, so I'm hoping that you have understood the session and you should be able to answer this. Yeah. So everybody agrees with option A, that is secondary antibody. So you saw the same way how you saw in ELISA, the primary antibody is detected by the secondary antibody, and the secondary antibody will have the enzyme which will give the reaction. Similarly, in Western blotting also, once the primary antibody binds to the membrane, you can add the secondary antibody that will bind to the primary antibody, and then you can add the substrate to see the reaction. Moving on to the next question, question number 18. Which of the following is a common application of agglutination? Option A, blood typing. Option B, pregnancy tests. Option C, detecting bacterial infection. And option D, all of the above. So I have discussed about agglutination. So if you remember, both direct and indirect. In this question I am asking about both direct and indirect. What is the common application of agglutination in general? Like, do not answer specifically for direct or indirect. In general, agglutination can be used for which of the following things? Blood typing, pregnancy tests, detecting bacterial infection, and all of the above. Okay, I have one answer, option D. Okay, anyone else wants to answer? Yes, so two answers now. Option D, that is the correct answer, all of the above. So agglutination, I have told you, it's a basically antigen antibody reaction, right? Antigen antibody forms a clump that will basically form agglutination. That agglutination principle can be used for checking the blood type. So all of you must have gone through blood test once in your life where you have checked your blood group. So to check the blood group, it's a very simple process where they take a drop of blood. So I have a slide. Yeah, so you have a slide, glass slide. Prick uh, prick your fingers gently, and then put the blood drop in 4 places, and then what they do is they add different types of antibodies. So they add the antibody against the A antigen, antibody against the B antigen, and antibody against the D antigen, that is the Rh positive or Rh negative antigen. So that is the antigen against the Rh positive factor. And then you have the 4th drop, that is for control. So if addition of anti A gives agglutination and addition of anti D gives agglutination. So what does it mean? That means in the blood antigen A is present and antigen D is present. That means it is an A positive type of blood group. Then if addition of uh, antigen A is giving an agglutination but D is not giving agglutination, that means A negative is there. For O positive, you will not see any agglutination in A or B, because in the O blood group type there is neither A nor B antigen is present in the blood. So that's it's called O. It's the blood group where you have none of the antigens present. Okay, if you have both the antigens present, then it will be AB type blood group. Depending on the D, you will have either plus or minus. Okay, so I am assuming this you must have seen also if you have gone for blood group testing ever. So it's a very common test and it can be done in any small laboratory. So it's interesting to see the blood getting clot, forming clumps like that, and you can actually check your blood group. Okay, so that is blood typing. Other than that, we had pregnancy tests, right? So pregnancy tests also, if you have ever seen a pregnancy test kit, a photo or a video, so in that you will see you can put a drop of urine sample and then you can get color. So that color is also a antibody antigen reaction. So in case of pregnancy, you have HCG antigens in your urine sample. So that the kit that is available in the market, those kits have antibody against the HCG factor coated on that strip. Okay, now once you add the urine sample diffuses through the strip. So it's like uh, the immunodiffusion assays that we were studying, right? In the slide, you added a drop of antigen in one well, so it was diffusing in the and it was diffusing through the agar. So similarly on this strip also, when you put the urine sample diffuses, and along with that, if the antigen is present, it is diffusing. If the antigen is present, it is going to react with the antibody and you are going to see a color band, because they have added some other components that can give a color. So you basically, the antigen and antibody reaction will happen. If the reaction is happening, you will see a band. If the reaction is not happening, you won't see a band. So that's how the pregnancy test, it's based on this immunodiffusion agglutination process principle. uh, Similarly, for detecting bacterial infection, I told you, like if we add a bacterial or viral agent to the RBCs, the indirect hemagglutination reaction, that is based on this only. Mostly the indirect hemagglutination reaction, it is used for influenza virus infection detection, but you can detect any kind of bacterial particle also using the same technique. So all of these techniques can be used, like can be a common application of agglutination. So this is the last question of the session. So anyone has any doubt in any of the questions that are discussed, or any of the techniques that have been discussed, or any other doubt from this topic altogether? Everything is clear to everyone? If anybody wants to ask something, you can ask me now. No one wants to ask anything. So I'm hoping the whole topic is clear to you. While solving the questions also, if you face any difficulty, I'll post the recording of this whole session, and you already have the lecture videos. Go through those. It is not a very complicated topic. You will get some idea. You will get the answers to your questions, to your doubts. If you have any doubt right now, you can ask me. I can surely try to clarify your doubt. Okay, if nobody wants to ask anything, then I think we can end this session now. Everything is clear to everyone, right? Okay, great then. Thank you all for joining. I'll end this session now, and I'll see you next Tuesday. Thank you.